I want to do today is I'm going to tell you some details about the format of the exam. Details of the format of the exam. Then I'll go through a review session that I put together based on questions that I got asked. And I'll ask that you don't ask questions during that period so I can make like a clean recording for the group. Then I'll open it up and you can ask questions, you can chat, or you can unmute yourself and ask a question. Okay, so let's first talk about the format of the exam. Your exam has 45 multiple choice questions that you will have 35 minutes to answer. The multiple choice questions are shuffled and they appear one at a time and they lock after you answer the question. This is to prevent people, if you happened to be communicating with someone else while you were taking the exam, you wouldn't be able to help each other because you would each have one question in front of you and it would be a different question. This again is to make it an independent exam. They do lock after you answer it, so you can't go back. So do your best, give it your best guess. Okay, 45 multiple choice questions, 35 minutes. For this section, those questions will open up at 10:10 and will close at about 10:50 once you start, the timer starts with those 35 minutes. You will then download the short answer document and I'll go onto Canvas, I'll show you where all that is. And you will have, it should only take you about 10 minutes for 2 short answer questions. I believe I have it open from 10:35 to 10:55. So assuming you used your 35 minutes, you would have 10 minutes. But if you finish the multiple choice early, you have a little bit of an earlier start you could take for those short answer questions. And I'll show you on Canvas where all of that is. The idea behind the exam is that I'm not going to make you pay for proctoring or online proctoring. In-person proctoring is no longer a thing, but online proctoring costs money, and I'm not going to make you do that. So instead, I made the exam so that I'm assuming you're going to study like you normally would, and you're not going to use any resources when you take the test. So you're just going to open up your laptop or your tablet, take the exam without any notes in front of you, without Googling things, without working together. So it'll be a normal, independent exam. When you submit the short answer questions, I put them through VeriCite, which is a plagiarism detection software that will tell me if you copied and pasted anything from the internet, which I'm going to assume you will not. You will take it from your brain. Okay, let me show you where all this stuff is on Canvas. So if you go to your Canvas page, again, this is your home page, scroll down. A reminder, the muscle worksheet is due in 45 minutes. You should do that. I also put up this review session right here. Okay. If you go to assignments, you will see coming up again that muscle worksheet due at 11, answering those muscle questions due tonight at midnight. This will be available starting at 10:10. Click on this. It's currently locked. Oh, I opened it at 10:05. Okay, so 10:05, you can answer that. You'll do that for 35 minutes. Then you'll come here, you'll do your short answer. It's locked until 10:35. You will answer that. That one, again, do at 10:55. Okay, that's going to be your test. The other thing I want you to bring your attention to is in the quizzes tab. I put up a technology survey that's just going to tell me, have you been able to use WebEx? Is it helpful? Have you not been able to use WebEx? Has it not been helpful? Have you just looked at YouTube? I just want to get a better idea of where everyone is, okay? So that is going to be how we do that. Okay, with that being said, are there any questions about the format of the exam? So please put them in the chat. With that being said, I'm going to now switch gears and I'm going to do a short review session. It's been a day. A short review session. Okay, let's share content. Okay, PowerPoint here. And again, this I posted. So Your exam covers chapters 5 and 6, bones and muscles. Are you going to post this recording? Yes, I will. I will post this recording too. A fun thing is happening from the instructor end of WebEx. When we do the WebEx recordings, they go onto the WebEx.com's like server system. And then when I first started doing this, like last week when I was practicing and on Monday, I was able to download the videos within about 10 minutes after class ended and I could post them to YouTube. Yesterday, the video Wednesday afternoon's video took about 20 hours to do that as opposed to 10 minutes. So I may not be able to post it like immediately, but I will post it as soon as I can. Yeah, I will. I will post it as soon as I can. Okay. Let's talk about the review session. Bones and muscles. I'm going to go just based on the questions I have, and then I will open it up for questions at the end. So I got the majority of the questions, and the thing that most people wanted to talk about was muscle contraction. And there were a variety of different questions that I think all go under this umbrella of muscle cell contraction. How does a muscle cell contract? How does a muscle fiber contract? Remember, cells and fibers are synonyms. How do motor neurons signal muscle cells? Why are the bands of the sarcomere important? Why is the sarcomere important? How do we label the sarcomere? What does acetylcholine or ATP or acetylcholinesterase or calcium do in muscle contraction? So I'm going to hopefully answer all of those questions here first. So keep in mind, a skeletal muscle cell is also called a skeletal muscle fiber. Those are synonyms. And it contains a plasma membrane that we call a sarcolemma. The cell has in it a few key things. The major thing you would notice if you were to look under a microscope at a skeletal muscle cell is that it's filled with cytoskeleton. We also call those cytoskeletons elements myofilaments. And what you would see is that they alternate dark and light bands. They look striated or striped. And the dark bands we call A bands, and the light bands we call I bands. As we know, because we now understand the structure of a sarcomere, the dark bands are dark because they're full of myosin, that thick filament. The thick filament makes the A bands dark, and the light bands are light because that's where you only see actin. So the light bands are light because that's where you only see actin. Myofibrils are made of contractile units called sarcomeres. Why are the sarcomeres important? They are what does the contraction of the muscle. So they are what contracts. And what we mean by contraction is when the filaments will slide past each other and the two Z discs will come together. So a sarcomere is measured Z disc to Z disc. It has A band in the middle, and again, those I bands in the middle of the whole thing we call the H zone, and there is an empty area called the M line where you won't find any actin. Myosin is in the middle, actin is anchored to our Z discs, and that's shown here. Again, thin filaments are actin filaments anchored to the Z disc. At rest, when the muscle is not contracting, there is no actin in that H zone at the middle of the sarcomere. The thick filaments are myosin. They're what do the action of muscle contraction. Inadvertently rhymed. Okay. At some point, make sure you can draw a sarcomere. And I'll talk about how the muscle contracts. Keep in mind our vocab words: excitable, contractible, extensible, and elastic. The muscles need to be stimulated by nerves. Skeletal muscles are voluntary. So they need a signal from your brain that says, hey, contract. It's time to contract. That's by our motor neurons. One motor neuron can stimulate several muscle cells, and we call that a motor unit. And they meet at an area called a neuromuscular junction, where the motor neuron will release acetylcholine into the gap, which we call the synaptic cleft, and the acetylcholine will bind to that muscle cell. The motor neurons come from the spinal cord. They come from the spinal cord, and they go and they innervate or make junctions with all of our skeletal muscles. They're going to release acetylcholine. Acetylcholine, again, is that chemical. We call it a neurotransmitter. And they release it into the synaptic cleft. Acetylcholine is released because of a signal that comes down the motor neuron. So the motor neuron will get a signal. It'll come from the brain down the spinal cord. That signal will cause calcium to fill the motor neuron. The calcium is what tells the motor neuron, hey, dump out your acetylcholine. And it will dump acetylcholine into the synaptic cleft, and that acetylcholine will bind to the muscle cell plasma membrane. What this does is it opens up a cation channel. Cation, remember, means a positively charged ion. Cats are positive things in your life. And there are two flavors of cations here that we're going to look at. The first is sodium. Sodium is our sprinter. So sodium is going to rush into the cell fast. This makes the cell have a positive charge now. We call this change in electrical potential, this change in electrical signal, a depolarization. So that rush of positive charge is a depolarization. It's followed by potassium. Potassium is our tortoise. It's a little slower. And it will eventually leave our muscle cell, creating this wave of positive coming in and then positive going out. Okay? If this moves down our muscle fiber, that is an action potential. So the acetylcholine binds to a cation channel. Sodium rushes in, making the cell positively charged, and potassium will leave, making the cell negatively charged. This is a depolarization. How does it end? Acetylcholine is broken down. It's broken down by an enzyme called acetylcholinesterase. We abbreviate ACHE, and this will end our contraction. And that positively charged wave, that depolarization, will spread like fire on a twig down our muscle fiber, down our muscle fiber. Call this an action potential, a flow of depolarization. Flow of depolarization. Okay. What does the muscle cell do in response? So the muscle cell is going to contract. The sarcomere specifically. The sarcomere is going to contract. And how it contracts is the myosin will reach up and it will grab the actin and it will pull it to the center. We call this a sliding filament model because none of the fibers are going to be destroyed or created. They're just going to slide past each other. They're going to contract. They're going to take up less space. And this sliding requires calcium. Calcium comes from an area in the cell called the sarcoplasmic reticulum, which is just a store of calcium. So the calcium is required because the myosin at rest is blocked from touching the actin. It wants to grab the actin, but it cannot. And when calcium comes, that releases the block and allows the myosin to grab the actin. And it uses ATP to do this. So the myosin needs two things. It needs calcium and it needs ATP in order to contract. Okay? So if we put it all together, we have acetylcholine being released from our motor neuron. Acetylcholine will bind to the sarcolemma. It will open up a cation channel. Sodium will rush into the cell. Potassium will slowly leave, creating a wave of positive charge called depolarization. That depolarization will move down the muscle fiber as an action potential, and the action potential opens up the calcium stores from the sarcoplasmic reticulum. Calcium is going to fill our muscle cell, and that calcium will release the block that is on actin and allow myosin to grab up and pull actin to the middle. The myosin will use ATP as the energy source for this, and it will keep going until either the signal is stopped by acetylcholinesterase breaking down acetylcholine, or the muscle runs out of ATP, which we'll talk about in a little bit. The muscle can run out of ATP. A lot of other people wanted to know about osteoblasts and osteoclasts, or other questions about bone growth, bone remodeling, the function of muscles on bones. All of that I'm going to put together here. So, ossification is the process by which we replace cartilage with bone. As an embryo and as a fetus, all of our skeletal system is present first as cartilage. And that cartilage needs to be replaced by bone. And the cells that do this are our osteoblasts. So the osteoblasts are going to cover the cartilage with a bony matrix, and it will dissolve and digest that hyaline cartilage, leaving the bone behind. So we then are replacing the cartilage with bone. At birth, almost all this is complete except for a few key areas. We have those fontanelles in the skull that allow the child's head to be kind of smushy so it can fit through the birth canal. And we also have our epiphyseal plates, or our growth plates, which are present in our long bones. The bones grow lengthwise from those growth plates. The cartilage is added to the top, and then on the bottom, or the distal end, the cartilage is replaced by bone, again by those osteoblasts, B for building cells. Those are our builders. They add bone. Bone remodeling is where the bone changes shape in response to something, whether that is muscle activity or hormones, okay? Bone remodeling is when that bone will change shape. And to do this, the bone is going to be reabsorbed or destroyed in some areas by our osteoclasts with a C, our chaos cells, and it will be added to other areas by the osteoblasts, our builder cells. So again, bone remodeling, the blasts are going to add bone, the clasts are going to destroy bone. If we get an increase in the circumference and the width of our bones, we call it appositional growth. And this whole process is controlled by growth hormones. When would we need to do this? So if calcium in the blood is low, your body does not care about your bones. Your body thinks in the short term. And so your body needs to maintain homeostasis of blood calcium, a set point, a happy medium for blood calcium. So if blood calcium is low, the parathyroid is going to release parathyroid hormone, which is going to stimulate those destroying cells, those osteoclasts, and it will take calcium away from the bones and put it in the bloodstream so that blood calcium is back to normal. Bones can also increase in size, and this is where the muscles come in. So we talked about with muscles, we learned about origins and insertions, right? Landmarks on the bone where the muscles attach. Those areas exert a lot of force. So the muscle is going to exert force on the bone, and the bone will need to get stronger and thicker to deal with that increased force. So if you are working out your deltoid muscles, you're doing some of those abduction, adduction exercises, right? You're increasing the force of the deltoid. That's going to add force onto your humerus, the insertion of the deltoid. And so the humerus is going to want to add bone to try to deal with that increase of force. And so we like to think that calcium tells us when we need to have bone remodeling. If calcium is low, bones are going to be destroyed. If calcium is high, we're going to add to our bony matrix. And the muscles tell us where. The muscles say, hey, we got a lot of calcium in the blood, put some over here. Come bring that extra calcium and put it here because this person, every day is arm day. And so the humerus needs more calcium. Okay. People ask about ATP. How is ATP regenerated? There are 3 ways that ATP is regenerated. Muscles need ATP to contract. However, there's only enough stored in our muscles for a few seconds of contraction. And so we have to make more. We have to turn adenosine diphosphate with a D into adenosine triphosphate with a T after we use it up. First, we can regenerate it directly. So our muscles can use something called creatine. Creatine phosphate has that phosphate group. It's stored in our muscles. We can take the phosphate from creatine phosphate and add it to adenosine diphosphate, making it adenosine triphosphate. We make 1 ATP for every creatine phosphate molecule. We don't need to use oxygen, but the issue is that store of creatine phosphate still goes fast, even if you're taking a bunch of supplements. So it's only going to give you 15 seconds of energy, approximately. You can also make ATP without oxygen. You can make ATP without oxygen. This is called anaerobic respiration. You can take glucose and make 2 ATP molecules. The issue with this is that you're going to build up lactic acid. This causes muscle soreness, and we use a lot of glucose for not a lot of return. The best way is to make ATP in our mitochondria using aerobic respiration. This uses oxygen. We get 32 ATP for every glucose, and we can go hours and hours without promoting muscle soreness. So that is how we regenerate ATP. A few people want to know about the pelvic girdle. What is the function? How is it different in males and females? The pelvic girdle is made of a few different bones. It's made of our 2 coxal bones, our hip bones, as well as our sacrum and our coccyx. Do not confuse the coxal and the coccyx bones. The coxal bones are the anatomical term for our hip bones, whereas the coccyx bone, that is our tailbone. It's in our butt. These are large, heavy bones. They're attached via ligaments, and they have deep sockets for our thigh bones, for the femur, to articulate. The function of our pelvis is to bear weight. These are big, strong bones because they hold a lot of weight. And for females especially, these are going to protect some very delicate reproductive organs. They're made of a fusion of 3 bones, the ilium, the ischium, and the pubis. Some key things you need to know. You need to know about the iliac crest that comes down into our iliac spine. That's what you can feel when you put your hands on your hips. The ischium is what is our sit bone. We have a tuberosity that holds the weight when we're sitting down. And the ischial spine helps form the narrowest spot in the hips for childbirth. The acetabulum is the deep socket for our thigh bone. When we talk about the measurement, we can measure wing to wing. This is our false pelvis. We can also measure the inlet, the true pelvis. And for comparing males versus females, you just want to think about childbirth. The female coxal bones, the female pelvis, is designed for childbirth. And so this means that the ilia are more laterally flared to create more space. There's a larger, more circular pelvic inlet. There's a shorter sacrum. The whole thing is rounder and has a larger pubic arch to create more space for childbirth. Whereas with males, it's the opposite, right? Their ilia are more vertically flared, they have a longer sacrum, they're less round, and their pubic arch is smaller. They have a smaller true pelvis. Okay? People want to know about muscle tetanus. So with muscle contraction, the muscle cell, the individual cell will either contract or not, but the whole muscle can have a graded response. So strength is related to the number of muscle cells that are contracted, and the characterization of that muscle contraction depends on the stimulation. So if we just get one motor neuron firing onto a muscle cell, that's going to create a muscle twitch. So everyone's had those eyelid twitches, right? Where you can feel that little bit of contraction, okay? The muscle needs to contract in a complete way, so a smooth movement. We call that a fused complete tetanus, a smooth movement. And that occurs when the motor neuron is firing rapidly on the muscle. If the motor neuron is not firing rapidly, either because of damage or some sort of injury is usually the case, that's where we can get unfused tetanus. And that is usually visualized by tremors. So if you look at Parkinson's patients, for example, they have a resting tremor, their hands are shaking. That's caused by abnormal firing from the motor neuron. So you want the motor neuron to fire very, very quickly upon the muscle to give us that fused complete tetanus. Increased frequency will lead to a smooth contraction, and again, force comes from the number of cells. A few people ask about muscle disorders. Muscular dystrophy, again, this is the inherited muscle destroying one. Duchenne's is the one I talked about. There are multiple kinds, but Duchenne's is diagnosed in early childhood, ages 2 to 7. It's usually fatal by the time the person's in their 20s or 30s because the sarcomere itself starts to fall apart. Whereas myasthenia gravis, this is diagnosed, onset is in adults and also fatal, but this is from a destruction of the acetylcholine receptors. So in muscular dystrophy, the sarcomere itself is falling apart, so the muscle can't contract. And in myasthenia gravis, the muscle is fine. It's just that it's not receiving input from the motor neuron because it doesn't have working acetylcholine receptors. Aging, muscle atrophy associated with aging, we call sarcopenia, sarco meaning muscle again. This is where we see a decrease in muscle mass, a decrease in overall body mass, decreased strength. But this is reversible. So you can maintain and rebuild your strength, the majority of your strength, if you maintain your exercise program. So again, muscles use it or lose it. I want to know about synovial joints. Synovial joints, again, you want to think of examples. So we have plane joints. These are in the intercarpals of your wrist. They allow you to spread your hands. And these are a sliding or gliding motion. These are non-axial. We have a hinge joint. The classical example here is the elbow. It's a uniaxial joint, can only move in one direction. Pivot joint. A pivot joint is also uniaxial. Think of the pronation supination that we see at our radial ulnar joint, right? So our arm bones can move in that pivoting motion around each other for pronation supination because of that pivot joint. Condylar joints. Condylar joints, we're thinking of our knuckles of our fingers. These are biaxial, so they can move back and forth and side to side, but they can't make a full circle. And this is similar to our saddle joints, but saddle joints can make a bigger circle. So think fingers versus the saddle joint is our thumb, the carpometacarpal joint of our thumb. We can get close to a full circle because of that saddle joint. And finally, we have a multi-axial joint. This is our ball and socket, our shoulders and our hips. They have a wide range of motions. Okay, so that is the end of the slideshow that I have prepared for you guys. Are there any questions people have? I'm going to stop the recording. Class is now officially